Okay, and our last speaker is Bing-Chu Wang, who will be presenting on factorized convolutional models for interpreting neuron-guided image synthesis.
Hello, everyone. Today, I'm going to talk about one piece of work that I've done through my PhD with my advisor, Carlos Ponce. And as many of us in this room have known, that regression method has widely used in this field of visual neuroscience. We have used it for predicting neural activities and have used them to synthesize images to control neurons or search for like functional regions or define new neuroscience problems. So this is like one side of the problems. So this is the mappings from images to the voxels or neural activities. And through the last few years, we have been very good at solving this regression problem from predicting neuron firings very well. And there's another side of this coin, which is the attribution problems. So that's the errors from neural activities to the images or any stimuli. So we want to know what type of features or what type of things in the inputs that are important to the neurons. So can we use our previous regression models to localize important features to neurons? So actually, this is a very common problem in both computer vision and visual neuroscience. In computer vision, people have tried to ask these kind of questions of, why did my model make such decisions? Like, why did it classify this cat as a cat? And what kind of features is the decision based on? And people develop interpretability methods like smooth gradient, grad chems, etc. And in visual neuroscience, we have other people asking other type of questions, but come down to the same underlying things, which is why does this neuron fires and where and what features does the neurons firing based on? And people have used things like receptive field mappings to answer these questions. In the computer vision land, people have these neural networks with inputs as images. And this neural network will process the images into some maybe scalar outputs, and people can pass gradient from this output to the inputs. This is like the gradient, and it will highlight some regions. If you do some things like smoothing, you highlight the region that has importance to these predictions. And sadly enough, our brain is not compiled with like Torch or TensorFlow or JAX. And so neurons doesn't pass gradient to pixel themselves, so we need to do something else. And usually what we do is to fit a neural network model of the neurons and then pass gradients through that deep neural network models. The issues that driving this research is that, is our DN models good enough to predict that spatial dependency or receptive field? And we think it's not enough in many cases. So this is a motivating study using an in silico setup. So since many cases we don't really know the neurons receptive field in vivo, we will first start with like in silico target neurons. And we hook up one unit in a deep network, and in this case, less than 50s, and then use another neural networks, maybe less than 50 robust, as the regression models of it. And then we will compute the gradients of the target units, like our model neurons, to the pixels as our receptive field, and then also compute the gradients from these encoding models, and then compare. So what are the common regression methods or linking procedures in this field? So in many famous papers, including BrainScore and like Colin Conwell's paper, people use this common workflow in neural regressions. Because we have too many features in this feature tensors in either transformers or convolutional networks, we will first do feature reductions, which usually involves like principal component analysis or sparse random projections. And then you apply certain penalized regression methods on top of it, and then predict your neurons. And how does that behave? So here, I will show this ground truth receptive field of that unit, of our target unit, so it's like the true receptive field in a sense. On the right side, I will show like the receptive field of the encoding models. As you can see, the predictive models, even with high predictive scores, can over or underestimate RF of the neuron by quite a bit. And to some extent, it's not super surprising because we basically vectorize or fatten the whole feature array and then do dimension reduction on it. So it disregard any source of spatial structures in it. And so if our regression method or encoding models doesn't correctly estimate the receptive field of the neurons, then how should we trust their prediction on something else? So that drives our development, our methods. We call it like factorized regression methods. So we desire this property of weights because we know neurons. One thing we know of neurons is that they have receptive fields, which means they have localized and also kind of continuous dependency on the pixels or the inputs. So we want the weights to kind of reflect that. We want the weights to be kind of localized and continuous. And so we made this our priors, and we made like a factorized weight tensor to be a factorized tensor, which means it has one factor is the spatial mask, telling us where does the neuron cares about, and one part is the feature vectors, which is what does it cares about. And this is definitely inspired by the David Klein's works on the neural system identifications and many of the Tolia's work. So how do we learn this system? Because it's kind of non-convex optimizations, so we need to initialize this factor really well. We use one use for priors, which is maybe the things that are correlated with neurons are the things that are useful for our neurons. So we pass the images through the brain, which is the monkey's visual systems, and also the convolution networks, and correlates the neurons' activations with all the voxels, all the tensor values in the activations. And then we factorize this covariance tensor or correlation tensors by NMI factorizations. And hopefully you'll get some good spatial mask and some good feature vectors. After that, if you want to refine your predictions, you can further refine the weights by like fixing one side of the weights and regress the other side, or fixing both sides. So what's the result? On prediction levels, so these are two tasks we've done. One is using evolution sets of images, and the other ImageNet images. And basically, the factorized methods doesn't show any edge, and that also doesn't has any flaws behind the previous method like IPC or SRPs. So it's very comparable. But the real edge comes from the estimations of the spatial dependency part. So if we now pass gradients through the factorized regression models, the receptive field estimate is much closer to the ground truth receptive fields, and it's much more accurate than the classic feature reduction methods. And that's especially the case if you fit the mask themselves in the post-regression steps. So given this, the methods, we applied them to one of our previous data on the neural guided image synthesis experiment. In this experiment, it's pretty interesting. There's a monkey sitting there and looking at the screens. And we have optimization algorithms propose images to the monkeys. And then the algorithms will gradually morph the images to increase the firing rate of the neurons. And you can see the post-stimuli times histogram, the PSDH here is keep going up, and also the firing rate of the neurons is keep going up. And in the end, the image seems to converge to something like looks like maybe parts of objects. But still, you can ask question, which part in this image does the neuron really cares about, and what is the real thing that's important? So we send this data into our pipelines, which is all the images through this evolution experiments, and send it through a convolution network backbones, and then do the correlations, and then factorize into 3 spatial masks and feature vectors. And feature vectors themselves are just numbers, are not super interpretable, so we applied the feature visualization methods to turn them into images, and these are the results. And you can also combine the spatial mask into this kind of weighted mask, which tells us which part of the image is more important. So here's one example. In this evolutions driven by the IT neurons, it ends up with these evolved images, and we do the factorization thing, and it now ends up with like 3 parts. One part it is a central object, which is this pink bowl thing, and under here is like this shades of this bowl, and up here is more the background part of this. And in combination, it will tell us the bowl is very interesting and important for a neuron. In contrast, if we do apply this pipelines with the evolutions for V1 images, the original end result of the evolved images is very complex, But this factorization will tell us basically the important thing is 3 parts: the central like gabor-like textures and some surround, and some others surround. And the real interesting part is very small actually. So we apply this to a series of data, and there's the original MEIs, or the most exciting images during the experiments, and you see these are complex images. And here is the composed spatial mask showing the importance of the features, and this time is highlighting maybe the eyes or maybe this node. And we can synthesize the MEIs from this factorized model again, which definitely simplified the images quite a bit. And this model can also predict the neuron firing rate pretty well through this experiment. Here are some more examples from IT visual cortex, V4 and V1. So you may ask, from this perspective, what is actually changing through visual hierarchy? So one thing we know is that actually our system has one free parameter, which is the number of factors you have. So from this learning theory, if you make your model very complex, you can regress more variance in your training set, but maybe have more test errors. So you have this kind of U-shaped curve in your train test accuracies. So maybe we can find the optimal factor number by this cross-validation approach. So we find that for V1 neurons, actually you just need around 2 factors, run 2 tensors to make correct predictions. For V4, it's 3.5, and for IT, it's much more than that, around 6 factors. And since this optimal factor number and complexity of the features are increasing along the visual hierarchy. Finally, so maybe this is just like a data fitting method, and neuron doesn't really care about it. So how do we really validate this type of models? And here I think this is one edge of this new type of models, which is if these factors are real, then we can manipulate them and ablate specific part of the model and then tell us does neuron still likes it or still predicts the neurons. So we will first perform this closed-loop evolution experiment and build the factorized model for neurons and then ablate some part of it and then show the images back to the monkey again. So here's one example. So this is one image evolved from this neurons, and this neurons, and we have our original models, and then we can mutate part of it, for example, the spatial mask part of it, and we can ask, does this neuron care about this specific spatial location, or it cares about just anywhere on this place? And we can put the features everywhere on the space or in the center of the space. On the other hand, we can also shuffle the features and do the same game. So for each of these models, we can visualize the content of them by feature visualizations and turn them into images. And so under each of one image is a distribution of images that highly activating each one of these mutated models, and we can show them back to the monkeys. And we find that, first of all, many of the one-factor model can correctly synthesize images that highly activated in neuron, matching the original activations. And if you spatially mutate the mask, it will decrease the firing rate, but not by too much, showing some extent of spatial tolerance, similar results for other factors. But if you totally mutate the spatial feature vectors, then the firing rate totally goes down, and showing that the feature selectivity is pretty high. In summary, we find that classic feature reduction methods and regression methods generally do not recover the spatial dependency of the neurons, and factorized regressions recover it much better. And we find that neurons in higher visual cortex require more factor-to models and suggesting a higher degrees of composition complexity. And these factorized models also create a small number of masks and factors, so we can targetedly manipulate these factors for hypothesis testing. And thanks for attention.
Thank you. We have time for one quick question.
That was a great talk. I'm wondering if you could gray out everything outside of the spatial mask, or I don't know, make the images even more simplified, and you think that they would still drive the neurons? I mean, the images you showed looked simpler, but there was still a lot going on in regions that I thought would have not been in the spatial mask.
Yeah, that's definitely a good question. I think the other compared method, maybe from Tolia's lab and DiCarlo lab, they they do a more constrained version of this. Basically, I think they apply the spatial mask in some sense in the pixel space, so they can constrain optimization in that space, and that may be a future direction. Here we kind of apply spatial mask more in the feature space, so it's like a looser constraint in the sense. Thank you.
We're out of time, so maybe you can follow up afterwards. Okay, let's thank our speakers again. Thank you.